Now I want to enter a different topic. It is known that antibiotics cause the death of bacterial cells. This was discovered in 1928 by Alexander Fleming, who observed that colonies of a bacterium, Staphylococcus aureus, that these colonies are destroyed by the mold Penicillium notatum, which produces a substance called penicillin. And the important thing I want to point out is that Penicillium notatum, this mold, is resistant to penicillin. And now we have many antibiotics, and the way they are tested is that they are placed on a petri dish with a bacterium, with a bacterial colony, and then we observe that some of them are very effective in destroying the bacterium, others are less effective because they have a smaller effect on the bacterium. We select the most effective. Now, how does resistance to these antibiotics develop in microorganisms? This is done through horizontal gene transfer. For example, from the mold penicillium. It has resistance to penicillin, and this resistance can be transferred to other organs. How does the horizontal gene transfer function? A cell may obtain resistant genes from other cells, from other organisms, by various ways. By conjugation, when the cells conjugate and there is a transfer of some genetic material. This can happen only between related individuals. This can happen through transformation. A cell will disintegrate and it is consumed by the living cell, but some part accepted or taken in from the remnants of the other cell is not consumed, but is adapted to be used by this new cell. So there is a transfer of a resistance gene to the genome of this bacteria. Finally, there is transduction, which is the transfer of a resistant gene by virus. We know that some viruses will move from one organism to another, and they can carry some genes, could be a gene for resistance. And in this way, one of these ways, the cell develops a resistance to a substance that is hurting it, and this can be achieved in nature by this horizontal gene transfer. It doesn't require sexual reproduction for the transfer of genes. Now, we have discovered all this, and now we know that we can use viruses as carriers of genes from one cell to another. And we are doing this in biotechnology. We are achieving GMOs, the genetically modified organisms, which are modified in such a manner that we take some gene from one organism and transfer it to another with the help of a carrier, the carrier being a virus. This is a big technology that is developing very quickly now, and we have learned how to do it, and we are employing it, but it also happens in nature. And when we have found a population of a bacterium which is resistant to an antibiotic, it has received its resistance through horizontal gene transfer from other organisms.